Half of chapter 15, we're talking about mutations in the first half. And here we want to talk about the fact that, as we've already discussed, these mutations can lead to disease, right? So if you have, a loss of function mutation in an important gene, for example, we discussed sickle cell anemia, that can lead to disease. I think I gave you cancer as another example. Um, so I want to discuss some of the methods that are used to screen for some of these mutations and some of these genetic diseases, and then talk just briefly about what could be done about them, if anything, in certain instances. So genetic screening would just be defined as using tests to try to figure out if somebody has a particular genetic disease, or at the very least might be predisposed to one of these diseases. And there's really two main types of screening. So the first type of screening would be to screen for a particular phenotype, basically, looking to see, do they have the symptoms of that disease, or even at a molecular level, is there some protein that's not functioning properly in their cells or in their body. And then the second type of screening would be to actually do some DNA testing and look to see, can we identify any specific mutations in a particular gene. If so, we may know, okay, if you have this mutation, then that's going to cause this particular disease. Let's talk about screening for a phenotype first. Uh, so, as I said, that could involve actually looking at your phenotype, your appearance or something in terms of your gross physiology, but it could also involve looking at the phenotype of a specific protein or a specific enzyme. So the example I've got for you here is a relatively common metabolic disorder that's caused by a mutation. This is called PKU. Um, that will basically reduce an individual's ability uh, to break down the amino acid phenylalanine. So the mutation is in this enzyme called phenylalanine hydroxylase. So if you have this disease, this mutation, that enzyme doesn't work anymore. You take in phenylalanine in your diet because we're eating protein that contains that amino acid. Your body can't break it down. It builds up and can cause serious issues especially during early neurological development, during childhood, and can lead to mental retardation and serious lifelong problems. So this is something that's just routinely screened for in hospitals now. As soon as a baby's born, they'll do a heel prick and get some little blood samples on a card, and they can use that for an assortment of tests. But one of those tests will be to look for the activity of this specific enzyme. So they can, in the lab, check to see if the phenylalanine hydroxylase enzyme is working from that blood spot. If it is, no problem. If not, uh, then we know, hey, this infant has PKU, and you can usually deal with PKU pretty well just by altering the diet. So just reducing the amount of phenylalanine in the diet to prevent that build up to toxic levels. And usually, as long as you do that during early childhood, then um, you're fine and don't have to worry about any of those neurological effects. So that would be screening for phenotype, whereas screening for genotype would actually be DNA type tests. And there's lots of examples of these, especially as DNA testing has gotten cheaper and easier to do over the years. Um, and I wanted to point out this kind of screening could be done at lots of different stages. So it could be done in adulthood, but a lot of these screens are happening, maybe in a zygote. So let's say you're having in vitro fertilization in a lab setting. You might have the fertilized zygotes genetically screened just to see if there's any serious genetic diseases before implantation. Um, You could do amniocentesis, where you're removing some of the amniotic fluid during pregnancy. And there would be some fetal cells in that amniotic fluid. You could get DNA from those and do some genetic testing. Or, of course, you could do it, after birth. And so just to give you, one example of how one of these DNA tests might work, and there's lots of different types, but this would be a fairly typical example using what are called DNA probes. And this particular test is for sickle cell anemia. So the idea is you would have your DNA samples from your individuals. In this case, it's 4 different individuals from one family. So mother, father, child, fetus. So each spot is a different person's DNA sample. And you would treat those DNA samples with these fluorescent DNA probes. So these are basically little short fragments of DNA synthesized in the lab that would have some tag added to them so they could be easily visualized by the researchers or the person conducting the test. And so you would have a probe that corresponds to the normal hemoglobin protein sequence, beta globin sequence, and then you would have a probe corresponding to the mutated, point mutated beta globin sequence that would lead to sickle cell anemia. And so simply all you would do is you would take these probes and basically wash them over those DNA samples. If there's a match in sequences, the probes would stick. If there's not a match, they would just get washed off and you wouldn't see them. And so what you would end up with is, in this case, mom and dad are fine. So you have just the normal probe would bind and the sickle cell probe doesn't. But if the sickle cell probe does bind, then you have a situation where, okay, that individual looks to be heterozygous for sickle cell anemia. And you could do, further tests to confirm, but that's going to be pretty convincing. So, as I said, lots of different ways to carry out these tests, but it's typically going to be either looking for phenotype or a DNA test to look for the actual genotype, look for those specific mutations. So if we find somebody does have a genetic disease caused by a mutation, is there anything we can do about it? And short answer is yeah, there are in certain cases there are. And there's typically 2 possibilities. So one would be modify the disease phenotype, which is basically just treating the symptoms in some way. Or the second possibility would be to actually try to go in and fix the defective gene through what's called gene therapy. And this would really be curing a genetic disease versus just treating it. So in terms of just treating the phenotype, the PKU example that I mentioned would fall into that category. So if you find an infant has PKU, you're not going to actually cure them of PKU. You're just going to restrict the amount of phenylalanine in their diet. And if you do that for a period of time, then they're usually in the clear and fine for the rest of their lives. Depending on the particular disease, there could be other approaches. Maybe you need to treat with drugs that could target specific proteins in some way, or maybe the mutation, is causing a missing or defective protein that could easily be replaced. So, for example, hemophiliacs that are missing specific clotting factors, those clotting factor proteins can actually be infused into the bloodstream periodically to give them some protection. But in terms of a true cure, you're talking about gene therapy. And the whole idea with gene therapy is you're actually going to go in and supply the cell with a correct version of the gene that does not contain that particular mutation. And there's really 2 big challenges associated with this. So it's pretty easy, pretty commonplace now to be able to synthesize in the lab a non-mutated version of basically any gene that you want. That's not really an issue. The challenge is how do you get that correct version into the cells that need it? So, for example, let's say you had a mutation that affected some particular liver enzyme gene. So you could make the correct version of that gene in the lab, but the challenge is how do you get it into those liver cells that need it? You can't just inject it into the bloodstream. It's not going to get necessarily where it needs to go, be taken up by those cells. Um, and then, on a subtler level, okay, if we do get the correct version of the gene into the cells, then how do you make sure the gene is being expressed at the correct level? So we don't want it to be overproducing the protein or underproducing the protein, because at that point, then you're still in kind of a gain of function or loss of function scenario. And so gene therapy has been successfully performed in quite a few cases now, and it has almost always relied on engineering viruses. Different types of viruses have been used, but one common type would be retroviruses. Basically engineering viruses to act as the delivery mechanism to help solve problem one here. We're going to use the viruses to get the correct version into the cells. And so the basic idea with this is you would have your normal version of the gene that you synthesized in the lab. You would insert that into your virus genome, um, put that virus genome into a virus particle that's probably been engineered so that it's not going to cause disease anymore, but is still capable of infecting cells. And then you have to introduce that virus to the cells that need that particular normal version of the gene. And there's two ways you could do this. You could either take cells, mutated cells out of the patient, and then mix them with the virus, in a laboratory setting in a petri dish, and then put the cells back in. Or in some cases, you might be able to just introduce the virus directly into the patient's body, and as long as the virus is getting to the cells in question, they may be able to do the job in vivo there. So that's all I've got for you for chapter 15. And we'll see you next time talking about regulation of gene expression in chapter 16.